Hello everyone, my name is Manish Kumar Yadav, and today's in this particular lecture we are going to talk about an overview of cellular respiration. So we are going to look at an overview of cellular respiration, and today's in this particular lecture we are also going to talk about glycolysis in a brief way. So let's talk about this. So in cellular respiration, what are the natural basic pathways that we are going to talk first? So when you talk about cellular respiration, first and very important pathway is glycolysis. First one is glycolysis. Second one is the conversion of pyruvate to acetyl coenzyme A, acetyl coenzyme A. Third one is TCA cycle, and the fourth one is ETS or electron transport system, in which we will be also talking about oxidative phosphorylation. So these all are the important steps that are involved in cellular respiration for the production of ATP molecules or adenosine triphosphate molecules. So first of all, we are going to see the basic steps of glycolysis. What are the enzymes which are going to incorporated in glycolysis pathway? How the glucose molecules are going to be broken down to convert the pyruvate molecules. Okay. So now we are going to talk about glycolysis. First of all, you need to understand that what is glycolysis. Glycolysis is a kind of metabolic reaction in which one glucose molecule actually can be converted into two pyruvate molecules. You know very well that glucose is a 6-carbon compounded hexose sugar. So if it is going to be broken down, so there will be production of two molecules of 3 carbon. So there is the production of two molecules of 3 carbon, that is pyruvate. One molecule is pyruvate, and so one glucose molecules can be broken down into two molecules of pyruvate. So that's what we are going to discuss today in this particular lecture. So first of all, we are going to see each of the steps of glycolysis one by one. So the first step of this glycolysis is that glucose is getting converted into glucose 6-phosphate. So as the name suggests, everything in biology, that if the name is going to be glucose 6-phosphate, it is going to meaning of that the 6 carbon of glucose is going to be bound with a phosphate molecule. That's why its name is going to like by glucose 6-phosphate. So how this phosphate molecule is going to be added? With the help of an enzyme which is known as hexokinase or glucokinase. So whenever there is a word of kinase, what does that stand for? Kinase means an enzyme which is responsible for phosphorylation, or it is going to transfer a phosphate molecule to a particular substance. So that's why the glucokinase or hexokinase comes under the category of transferase enzyme. That means it is going to transfer a functional group, that is phosphate. So hexokinase means a kinase enzyme which is going to act on a hexose sugar. That's why its name is hexokinase. Why we are writing it glucokinase? Just you need to understand that glucokinase is an enzyme which is only found in liver, not in all other cells. So kinase is found in all other cells. Glucokinase is found in the hepatocytes especially. So this is how we are going to deal with it. So glucose is a 6-carbon compound, and still we have glucose 6-phosphate, which is also a 6-carbon compound. That means hexose sugar is just being added with a phosphate molecule. So if there is an addition of phosphate molecules, from where this phosphate molecule is going to come in this particular reaction? So again, ATP molecule is coming for this donation. So ATP molecule will come and provide the glucose molecule 1-phosphate, and it will be converted into ADP, adenosine diphosphate, because 1-phosphate of ATP molecule is going to be involved with the conversion of glucose to glucose 6-phosphate. So this is the first step. Then what about the second step of it? In the second step, you will see that glucose 6-phosphate will be converted into fructose 6-phosphate. Fructose 6-phosphate. So you need to understand that glucose 6-phosphate is actually an actually aldose sugar. It is an aldose sugar because there is an aldehyde group. It is an aldose sugar. Glucose. I'm talking about glucose, not about glucose 6-phosphate basically. So aldose sugar is a glucose sugar. Because there is an aldehyde group in this glucose. And what about fructose sugar? Fructose sugar is a ketose sugar because there is a ketone group. Okay, so it is a ketose sugar. Another word which can be used for glucose is that glucose is also known as pyranose sugar, because there is a pyron ring. Pyron ring is a ring which has total 6 carbon that are going to play a role in the formation of a ring. And in the ketose sugar, especially I'm going to talk about fructose is a furanose sugar. So there is a furon ring that is found over here. So glucose 6-phosphate is going to be converted into another isomer of it, that is fructose 6-phosphate. Okay, that's why an enzyme which is coming here is a kind of isomerases. Okay, so the class of an enzyme is isomerase, and whenever this is going to play a vital role in the conversion of glucose 6-phosphate, that's why its name is phosphohexose isomerase. Its name is phosphohexose isomerase. So this is the name of it, phosphohexose isomerase. This is the second step of this glycolysis. Now let's talk about the third step of it. What is going to be happen with this fructose 6-phosphate? Now the fructose 6-phosphate will be converted into fructose 1,6 biphosphate. Fructose 1,6 biphosphate. What is it? Fructose 1,6 biphosphate. Fructose 1,6 biphosphate is nothing but there is an addition of another phosphate molecule on this fructose 6-phosphate. That's how there are two phosphate molecules in this fructose 1,6 biphosphate. So from where this again phosphate group is coming, phosphate functional group is coming? So again this phosphate is coming from ATP molecule, ATP, and again ATP will be converted into ADP because the phosphate molecule or inorganic phosphate is going to be utilized by fructose 6-phosphate. So that's why the fructose 1,6 biphosphate we can get from here. So fructose 6-phosphate will be converted into fructose 1,6. What is an enzyme for this? Which enzyme is going to catalyze this particular reaction? Enzyme is PFK, phosphofructokinase. Phosphofructokinase is also known as base battery of glycolysis. It is called as base battery of glycolysis. So this is how these 3 steps are very, very important for glycolysis. Now we are going to talk about some more steps. So let's talk about that. Now this is the 4th step we are going to talk about. So let's talk about the 4th step of it. We have fructose 1,6 biphosphate. Okay, so this is what we have, fructose 1,6 biphosphate. Now fructose 1,6 biphosphate is going to be broken down. So step is known as cleavage because there is a complete broken down of fructose 1,6 biphosphate into 2 basic compounds that is going to be of 1 molecule of 3 carbons. So that's why fructose 1,6 biphosphate will be having with 6 carbon. There are total 6 carbon. So when there is a cleavage of this 6 carbon, that means we are going to get 2 compounds which will be having with 3 carbon and 3 carbon. So there are 2 molecules of having with 3 carbons of each. So now what we are going to get from here? There are 2 molecules. First one is DHAP. And second one is G3P. What does it mean by these 2 molecules? THAP dihydroxy acetone phosphate, G3P glyceraldehyde 3-phosphate. Again there is a factor of keto sugar and aldose sugar. This is basically a keto sugar, and this is basically an aldose sugar. So now you know very well that glycolysis is a process in which all the things are coming from glucose. That's why this keto sugar must be converted into aldose sugar. So now THAP will also be converted into glyceraldehyde 3-phosphate. This is again going to be conversion of an first isomer to second isomer. So again the isomerases will come here, and the isomerase is going to be triose phosphate isomerase, triose phosphate isomerase. So this is an enzyme which is going to play its role. Now fructose 1,6 biphosphate has gone into the cleavage. Why? What is the enzyme which is going to catalyze this particular reaction? Enzyme which is incorporated in this particular reaction is known as aldolase. This is aldolase. Aldolase, an enzyme which is going to cleave fructose 1,6 biphosphate into two molecules. First one is dihydroxyacetone phosphate and second one is glyceraldehyde 3-phosphate. So this is the fourth step of it. Now let's talk, I told you that dihydroxyacetone phosphate has been converted into glyceraldehyde 3-phosphate. So now we are going to see all the further steps with the help of this glyceraldehyde 3-phosphate, not with dihydroxyacetone phosphate. So now let's talk about the next step. Next step, what we have? Glyceraldehyde 3-phosphate. Glyceraldehyde 3-phosphate. We have glyceraldehyde 3-phosphate. Now this glyceraldehyde 3-phosphate is going to be converted into 1,3 biphosphoglyrate. 1,3 biphosphate glycerate. So glyceraldehyde 3-phosphate is having with one phosphate molecule, and that is going to be present on the 3rd prime carbon. So how this glyceraldehyde 3-phosphate will be gaining with one more phosphate and be converted into 1,3 biphosphate phosphoglycerate. So again there is no ATP which is coming for this. The inorganic phosphate which is present in the cytoplasm is going to help in this particular step, and this inorganic phosphate is going to be merged with this glyceraldehyde 3-phosphate. And very important step is here, that NAD+, which is a coenzyme nicotinamide adenine dinucleotide, will be converted into NADH. So just remember this. Glycolysis is an oxidation. All the steps of glycolysis, all the steps whatever I showed to you, that all the steps are an oxidation reaction. All the steps are oxidation. There is an oxidation of glucose molecule, but NAD+ is getting converted into NADH. This is a reduction of NAD+. This is a reduction of NAD+. So now NAD+ is a reduced coenzyme. This is a reduced coenzyme. Okay, so NAD+ has been converted into NADH. That means there is a reduction of NAD+ molecule. Now we have 1,3 biphosphoglycerate. What is the enzyme which is going to catalyze this reaction? The enzyme which is going to convert this glyceraldehyde 3-phosphate into 1,3 biphosphoglycerate is glyceraldehyde 3-phosphate dehydrogenase. So whenever you hear this particular word, that's a glyceraldehyde 3-phosphate in metabolism, you need to understand that this is going to reduce NAD+ or FAD+. That's why its name is dehydrogenase. So why we have now 1,3 biphosphoglycerate. Now the next step is what? The next step, 1,3 biphosphoglycerate will be converted into 3-phosphoglycerate or 3-phosphoglyceric acid. Now there are two phosphates in this 1,3 biphosphoglycerate. First phosphate is on the 1st prime of carbon, second phosphate is on the 3rd prime of the carbon. So now one of the phosphate molecule is going to be utilized, and which one phosphate molecule? The phosphate molecule which is present on the first prime carbon, it is going to be devoted to ADP. Now ADP, which is present in the cytoplasm, will get this particular pi and it will be converted into ATP. Okay, so now by the first time in glycolysis we are seeing that now we are going to get ATP molecules. So what this is the substrate level phosphorylation, first substrate level of phosphorylation. We can say that this is the first substrate level phosphorylation. Okay, so this is the first substrate level of phosphorylation. Why we are calling it a substrate level? That means substrate which were used, that was glucose, which converted into 1,3 biphosphoglycerate by various steps, now is going to devote one phosphate molecule from its part and it is going to use in the production of ATP molecules. So now we have 3-phosphoglyceric acid. Enzyme which is responsible for this particular reaction is PGK, phosphoglycerate kinase. Next step, now 3-phosphoglyceric acid will be converted into 2-phosphoglyceric acid. That means the phosphate molecule which is present on the 3rd prime of carbon, it is going to be shifted on the 2nd prime of carbon. Okay, the enzyme which is responsible for this particular reaction is phosphoglyceromutase. So this is another step which is again very important. Now 2-phosphoglyceric acid will be converted into phosphoenol pyruvate. Now we have phosphoenol pyruvate. Enzyme which is responsible for this particular step is Aldolase. Aldolase is responsible for a deduction of one water molecule. So now we have one water molecule from here. One water molecule we can also get from here. So now PEP, phosphoenol pyruvate, we have. Now it's time for the last step of glycolysis. In the last step, PEP will be converted into pyruvate. So now we have pyruvate molecule. The enzyme which is responsible for this particular step is pyruvate kinase. Pyruvate kinase is the same enzyme which is going to utilize for gluconeogenesis as well, and the pyruvate kinase is also getting used in this particular step. Pyruvate kinase, that means it is again going to do the phosphorylation, and phosphorylation is of an ADP molecule which is present in the cytoplasm. ADP will be converted into ATP. Now that there is a second substrate level of phosphorylation. Substrate level of phosphorylation. So now you can see that there are all the 10 steps of glycolysis has been completed. Now we are going to count the ATP and NADH molecules in these particular steps. Now you can see that first ATP molecule we utilized we invested actually in the conversion of glucose to glucose 6-phosphate. So just remember it, glucose to glucose 6-phosphate. Here we used it, glucose 6-phosphate. This was the first ATP molecule we invested. Second ATP molecule we invested where we converted fructose 6-phosphate into fructose 1,6 biphosphate. Fructose 1,6 biphosphate. Here we utilize second ATP molecule. Now you can see that we are total investing how many ATPs? 2 ATP molecules we are going to invest in glycolysis. Now this is the complete investment. This is the investment actually in a one glucose molecule. Investment. This is the investment. Now it's time to get ATP molecules. So first ATP molecule we got from First ATP, where we got first ATP molecule when there was conversion of 1,3 biphosphoglycerate into 1,3 biphosphoglycerate into 3-phosphoglyceric acid. So first phosphate molecule we got here. Then the second ATP molecule we got when there was conversion of PEP, phosphoenol pyruvate, into pyruvate. Then we got the second ATP molecule. So how many ATP we have got? 2 ATP from 1 glyceraldehyde 3-phosphate, So I told you there are 2 glyceraldehyde 3-phosphate dihydroxyacetone phosphate was also converted into glyceraldehyde 3-phosphate. So 2 glyceraldehyde 3-phosphates are going to have the steps together. That's why we can get total 2 ATP. That means 2 times, that means we are going to get 4 ATP. So how many ATPs we actually are gaining from this particular glycolysis of one glucose molecule? So 4 ATP we got from one glucose molecule, 4 ATP, and total investment was of 2 ATP. So now we have total 2 ATP molecules we got gained actually. So this is only what we were looking for. We also got 2 NADH molecule, because when there was conversion of glyceraldehyde 3-phosphate into 1,3 phosphoglycerate, we got actually 2 NADH molecule as well. So again NADH is also important, because one NADH molecule will be having with 2.5 ATP molecule. So total we have 2.5 into 2, so we have total 5 ATP molecule, 5 ATP molecule from this particular step. This is only what we were looking at glycolysis? No, another thing which is also present here, that is pyruvate. Pyruvate is the thing what we were looking for glycolysis, because the main importance of glycolysis is for the pyruvate molecule, because once the glucose molecule will be converted into pyruvate will be converted into acetyl coenzyme A. Acetyl coenzyme A. Now acetyl coenzyme A will enter into TCA cycle, Krebs cycle, or citric acid cycle to create more NADH, FADH2 and ATP molecules. And then next is the ETS, electron transport system, in which we are going to use our NADH, FADH2 to produce ATP molecules. So this is the overview of this particular glycolysis and cellular respiration. In the next video we will be talking about Krebs cycle, TCA cycle, and next one we will be talking about ETS or oxidative phosphorylation. Thanks a lot for watching this video.